Hi, welcome to Social Research Insights. My name is Kamakshay Musnuru. I am Principal Consultant at Social Research Insights. Social Research Insights is not a business organization, it is just a freelance training outfit. We are just a couple of individuals who are interested in providing training on data science and analytics for both academic and corporate needs. We have a lot of content online and all the content is free. However, we have a small request that you can consider us calling for technical talks for both faculty development programs or staff development, whatever it is like. This is in fact one of my video demonstrations which belongs to a series of presentations. They're related to a topic bioinformatics through R programming language. In my previous videos I've explained how to deal with obtaining the genomes or gene sequences from different databases, just like NCBI of US, EMBL of UK, DDBJ of Japan, something like that. I also explained how to obtain the gene sequence, right being inside the R programming language. This is actually the R programming language. RStudio, which is a front end for, it is an editor for the backend R console. The left bottom side we have the R console here. This is actually the R, where we communicate to the backend R programming language. RStudio has 4 important panes, and you can just find one of my other videos where I explained everything about this front end, back end, editor, language, everything. In this video I'm going to show you how to figure out a particular problem in bioinformatics, that is about finding similarities or differences between genomes or gene sequences. Recently I came across a couple of blogs where I happened to read about a fact that 60% of human genome is identical with banana, and 60% of the same human genome is identical with fruit fly, and 99.99 or something, I don't know, I don't remember exactly, but which is identical with chimpanzee or baboon, something like that. One important fundamental fact that it might not be possible for us to come up with a hypothesis that just because 99.99% of human genome is identical with chimpanzee, that is why chimpanzees are our forefathers, or we are descendants of, I don't know in which way that makes sense, but this type of analysis, like correlation, associations, and similarities, differences, might be useful in order to figure out a fact about the common characteristics of any two given species, just like apple, banana, or maybe custard apple, pineapple. So they all belongs to a particular the category of species. And when we come to this all the creatures belongs to one single category, that is a living organisms. We all living, all these plants, quadrupeds, bipeds, and millipedes, whatever it is, pedal oriented creatures. We all belongs to one particular category called life. So perhaps the life depends on some essential inputs, and few of lives has sense organs. And anyway, I'm not interested in the philosophy of life and emergence and evolution of the theory of biology, the evolution in biology and all, etc. There are people to take care of that. And in this video I'm going to show you how to find the similarities between two different species. I've chosen two gene sequences, one related to banana and the other related to apple. And here, if you want to get the data data related to gene sequence of banana, you just have to go to the NCBI, or you can also go to EMBL, but I prefer NCBI because I don't know, right from the beginning I'm working with NCBI. You just have to choose this nucleotide here in this button. You just have to scroll down and find the nucleotide somewhere and ensure that there is nucleotide. And here banana, the nomenclature, the scientific nomenclature for banana is the Musa acuminata, and just type here and you'll get so many. You see how many links we get. We have such a many possibilities too. There are different alternatives too. Anyway, there is one particular important ingredient in this search result, that is accession. Every gene sequence will be having a valid accession number, and this number is very, very important for us. Anyway, I have another way of searching for the. I'll go to the browser, and there I go for a complete genome of Musa acuminata NCBI. That will give me a link, complete genomes, and there I'll find the accession numbers. I think these are a bit more reliable than just going to NCBI and there trying to, because I'm not basically from bioinformatics. I know very little about these gene sequences, and I'm basically a mathematician. I think this is the right way to, because we have different varieties of banana here. I also found a phrase here, Malaysian banana, something like that. There are 4 different varieties: Musa acuminata, subspecial Melex, and this is I don't know how you pronounce. Musa balbaceana, Musa itinerans, Musa textilis, or whatever it is. I don't like to waste my time. Okay, you can choose one of the accession numbers. Now this time you can go for this accession number, or you can just simply click on this link, so that you will reach to that particular site where you find this is very, very important. This is FASTA. You find some link called FASTA, and we need this FASTA file. You can also alternatively go here and press the file, and you can choose FASTA here, create file. You will get the file ready for the download. And once you get the file ready, you just try to save that in one of the folders in your computers. Here I'm going to show you where I have CD. I have that in downloads. In downloads, I think I have genomes, CD, genomes, ls. So here I have I think I saved it as banana sequence FASTA. So we need this .fasta format file from the, why? Because in our programming language we have one important package, secinr. You can find this package from CRAN. CRAN stands for Comprehensive R Archive Network, which is an official repository for most of the packages. I mean, approximately there are 10,000 packages available for, apart from the bioconductor and other RForge, etc. You can just if you press CRAN, you will get the Comprehensive R Archive Network, and where you can find the package secinr. So once you go to there, you see here, right side, you get the packages by name. Say go for the find secinr. You see here, biological sequence retrieval and analysis. Anyway, I'm not going to use this package for entire analysis. I just want this particular function, read FASTA, to read the FASTA files in which we have gene sequence, and those files are saved in one of the. So how you are going to do that? I'm just going to create an object called malice, which means malice is for apple. You can download the gene sequence for apple in the same way. The apple is known as Malus domestica. That is a chemical name for apple. I mean, scientific nomenclature to refer to apple. First, let me show you the banana. I don't call banana sequence because in that FASTA file we are going to have a hell lot of information, and gene sequence is one of the ingredients. So read FASTA, but ensure that you use this command, library secinr. Once you type this, you just have to press enter, control enter in RStudio, where the library secinr is available for data analysis. Now I'm going to show you how to import. You can just write banana and then read FASTA. Don't write the entire command, you just have to use tab function. And now you can go for the absolute path, just like this, downloads genomes and banana sequence, something like that. So you'll get that into the banana. If you're not if you're unsure about this writing all this path, you just can use the shortcut file choose and then press enter. You'll get the unity file Explorer in Ubuntu, go to the documents. I think I should go for the downloads, genomes, in which I have banana sequence. So either way, anyway, it is good. If R is silent, that it means that it had done some work for you. Now in banana, we have all this information. If you go for head, banana, then you get some almost all information, whichever is there in the. This particular ingredient is the element that which we actually is in search of. This is what is the gene sequence, and we don't want all this meta data about this gene sequence. Now what we try to do, banana, and of course we don't put dot because dot has special significance in object-oriented programming. Anyway, R is not object-oriented. It is, but it's mostly functional programming language. Banana sequence, and go for banana, and now dollar string. This is what is called splicing in our programming culture. Whenever you put this dollar string, then R try to show you what are the different elements available for your analysis. Then press tab and then enter. Now go for head, banana, sequence. Just use tab buttons every time. Now you see we just have sequence in our user created object, banana sequence. This is just created by me, that is why I call user created object. Now in the same fashion, just go for malus, which means apple. Go to the downloads, genomes, and then go for the malus sequence one, any one of it, doesn't matter. Then go for malus, and then sequence. This is the left assignment operator in our programming language. This is going to be malus, and dollar string, which will let you know where the sequence is. Now head, malus, sequence. Now I say TGT, so these are the nucleotides, the basic information about the gene sequence, base pairs or whatever you call that, Now let me see how many. What is the exact information about how many nucleotides are there in banana? How many nucleotides are there in malus? Something like that. For that, we have a wonderful function in R, that is table. Banana sequence. You see there are 235 A's. This is adenine, cytosine, guanine, thymine, something like that. These are the base letters in the gene sequence. There are 335 A's and 153 C's, 123 G's and 176 T's. And now same fashion, let us go for the malus. You just have to press upper arrow key in your keyboard to go for the history, or just before. What is the level of history there? You can just get back the previous statements. Malus sequence. Now, this has additional information: A, Z, G, K, M, N, O, T, W, Y. I really don't know what it is. I even don't know whether this sequence is a correct sequence for the analysis or not, but I just got it as in random fashion. But anyway, here we got a problem that in banana we just got 4 base letters, I think you call nucleotides. In malus you got A, Z, the apart from A, C, G, T, you got K, M, N, or W, Y. For search data, it is not possible for us to find the similarity using Pearson correlation coefficient. So now you need to know what are the different statistical techniques available for finding the relationships between these two gene sequences. We can go for a chi-square statistic, and that is meant for the tables like this. Chi-square statistic is useful to perform a test for cross tabulations in statistics. You can also go for Pearson correlation coefficient. In short, we call it as R coefficient, which is useful if you have numerical data given in 2 different columns. Each column is a vector, data vector, But here, if you go through this gene sequences, it is very clear to you that it is categorical data. There are 4 categories in banana sequence. There are more than 4 categories in apple sequence. Now, what we need to do? So we both of these technique, chi-square test or chi-square statistic, and as well as correlation coefficient R, is not useful for us. So I figured it out through a particular methodology. I first try to find an F statistic instead of chi-square statistic and coefficient R. I convert that F statistic into correlation statistic, which means the coefficient R, by using statistical theory or the expression R is equal to F divided by F plus, here I have that, F divided by F plus degrees of freedom 1 divided by degrees of freedom 2. And these degrees of freedom 1 and 2 are the 2 different degrees of freedom which were required to find the p-value for this F statistic. If you don't know all these statistics and the probability values, then perhaps you might need a basic course in order to understand all nitty-gritty or this statistics jargon. So you better go through a couple of or you can just throw yourself online to figure out what is this F statistic, chi-square statistic, or coefficient, or blah 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 blah. Now what I'm trying to do, I have a beautiful test called WAT test. Now I try to perform this test on both of these tables, and then I'll try to obtain the F statistic. And by using this F statistic, I figure out the R coefficient, and from there it is possible for us to detect whether what amount of similarity really exists in between the banana and Python. Now go for the table banana sequence, and then table malus sequence. Now beautiful, we got this F statistic here in the output, and that is, I think it is 0, right? Yeah. And degrees of freedom 3 for numerator, degrees of freedom 9 for denominator, and the p-value is, so this p-value clearly shows that the F value, whatever we have, is statistically insignificant. Okay, let us reverse the order and see what's going to happen. I think this 0 value is not going to help me in any way. So table banana sequence, yes, yeah, I think this time I have F statistics, so it is better to have malus at first and the banana, I mean, it doesn't matter whatever but the p-value is going to be the more or less same, 1.206 e to the power of, but with a 0 value we are not going to do any type of analysis, and that is why we just need so this is what is the F statistic. And now I created a function to convert the F statistic into R. That is very simple in R, creating a function, because R is known as the functional programming language. R from F, this is just username. We need to give one username for every function, and left assignment parameter, then I should tell R that this is going to be a function. Now what are the different inputs? We are going to supply 3 different inputs as arguments. First one is going to be F statistic, and second one is degrees of freedom 1, that is 9, and third one degrees of freedom 2, that is 3. Now once I'm going to return a particular statistics that is going to be square root of F divided by F plus the ratio of degrees of freedom over degrees of freedom 2, something like that. Once you finish writing this function, you just have to select the entire code and press Control Enter. Then if R is silent, then it does something for you. You don't need to worry. Otherwise R returns some red color letters that something went wrong in your formula. Now once you compile or execute this set of instructions as in the form of a function, then what you need to do is that you just go to the console, and there you check by pressing tab. If R returns the entire name of the function just like this, then R recognized your code as a valid function in the workspace. And now go for the I'll copy paste this, or you can also just write this here, and degrees of freedom 9, degrees of freedom 3 for denominator. Now you see this is actually the R statistic, the coefficient R. I'll call Pearson R coefficient, whatever it is. If you want to read what is the call Pearson coefficient R, then go to Wikipedia. There's nothing wrong. It is an open source knowledge management platform, and there's nothing wrong. Yeah. Correlation coefficient Wikipedia. Then you'll get a hell lot of information about the methods. You can just study. There are different methods like Spearman rank correlation, Kendall-Stowe rank correlation. This is what we are talking about. Pearson product movement correlation coefficient. You'll find the methods here. And additionally, you can also check for information as how to convert F statistic into R statistic, or correlation, something like that. You'll get a hell lot of information even for that. Okay, now let me go ahead. Now this is what is the R value, which is the correlation coefficient. From this value we can infer that the relationship is positive and it is very strong. 99, I cannot say 99.9%. I can only say it's close to 1, very similar, and it is positive. Because the R value range from -1 to +1. -1 is very strong but negative, +1 is very strong but positive. Now if you want to figure out how much of variance in malus is being explained by banana, then we need to convert into the R squared, which is also known as the coefficient of determination. I save this, I'll go just above, save this into R, then I call this as R square. It is coefficient of determination. Now it is possible for me to say that 99.99% of the gene sequence of apple is similar to that of banana, something like this. So though I don't say apple, neither malus is the descendant of banana, nor banana is the descendant of, that is a foolish statement, right? But 99 point, the characteristics in these two fruits, or maybe some life oriented characteristics in these two fruits, are 99.99% similar or close, something like that. We can also go ahead and do some statistical test whether this relationship is statistically significant or not. Anyway, that I will try to explain in my next. And I'm going to tell you how to, most of the time we are interested in the plots. When I am doing some statistical analysis, I might be interested in writing a paper or article or do some reporting purposes, where I wanted to compare these two gene sequences with the help of a couple of plots that might looks good. And for that, what happened, I just created a function, comparison plots. I called it as comp_plot, and it's again a function. And these two gene sequences, the sequence for apple, sequence for banana, are going to be the essential inputs for this function. And now this statements par or parenthesis mf row, this is the way we create the subplots in our programming language. This particular portion of the statement means that I want two plots in a single row, something like that. You can just go online or see my other video demonstrations to understand. And there is a beautiful function called bar plot, and we are using this method over the table of sequence 1 with some color coding, like col is equal to 1 to 4, which means take 4 first colors from the color palette, a default color palette in the R programming language. And I create the two plots, and let's say once you create this function, the same fashion, just press Ctrl Enter to compile that code. And now you go for comp_plot. Now you see R recognize this as a plot. Malus sequence comma, banana sequence. Press enter. Beautiful. So there's a huge amount of difference between these two, but that difference doesn't appear to be statistically significant to me. Anyway, I cannot come up with such a premature inferences. We need to do some statistical testing. Maybe we have to do the correlation test. Colbius correlation test is perhaps the right test for that. Since the value is very close to 1, there is nothing wrong for us to state that there is 99.99% of similarity between these two gene sequences. And whereas through the visual analysis, though there exist some differences, that is just because of the difference in the sample size, but not in the quality of the data. Anyway, that I will try to explain in my next video. And this is how you can just perform some statistical analysis in order to figure out some similarities or dissimilarities between two different gene sequences. If you like my video, please press like and subscribe to my video channel. Stay tuned for the new content. Thanks for watching.